everyone, and welcome to today's STERIS Tech Talk on practical navigation of the approaches contained in ISO 11137-2 and ISO 13004. Our presenter today is David Ballard.
Good morning, everybody. This is David Ballard, Senior Scientist at STERIS Laboratories in El Paso, Texas, USA. My co-author on this presentation is Sarah Conway. She is the Laboratory Technical Manager at the STERIS Laboratory in Tullamore, Ireland. Today, we will have a brief overview of the microbiological radiation validation and laboratory testing performed in the different phases of the validation. However, for a more in-depth look at the microbiological radiation validation phases, please visit steris-ast.com for other webinars covering this topic. We'll be reviewing the different approaches contained in the radiation standards 11137 Part 2 and 13004. Then we will discuss approach selection with working examples. The practical examples will demonstrate how different product requirements drive the selection and approach used for validation. And finally, we'll summarize key takeaways on the approach selection. While this presentation predominantly looks at the microbiological testing used to establish the minimum dose, it also touches on the importance of maximum dose testing. However, it does not look in depth on this topic or other aspects of the radiation processing or validation. For more information on these topics, please visit steris.ast.com. An additional note throughout this webinar, when we talk about sterility assurance, we'll be referring to a sterility assurance level, a SAL of 10 to the minus 6. However, it should be noted that other sterility assurance levels can be validated. There are some key definitions that we need to consider for this presentation. The first one being colony forming unit, or CFU. This is a unit of measure for microbiological assays, and we use it as an estimate of the number of viable cells in a given sample. Paired with that is a bioburden. It's the population of viable microorganisms, or CFUs, in or on a product or the sterile barrier system. SAL, sterility assurance level, is the probability of a single viable microorganism on an atom after sterilization. A 10 to the minus 6 sterility assurance level will give you a probability of 1 in 1 million chances of a non-sterile device. Some additional definitions include verification dose. This is the dose of radiations that's predicted to give a predetermined SAL. When we're looking at method 1 or method 2, that predetermined SAL is 10 to the minus 2. For the VD max method, it's predetermined to 10 to the minus 1. The sample item portion, or SIP, it's the defined portion of a healthcare product that will be tested. The SIP could be 1 or it can be less than 1. Sterilization dose. The sterilization dose is the minimum dose required to achieve the specified SAL, in this case a SAL of 10 to the minus 6. The maximum dose. It's the highest dose that can be applied without compromising the safety, quality, or performance of the product. There are some important phases that we need to consider in the microbiological radiation validation. The first phase being the test method validations. The second phase will look at the enumeration and characterization of the bioburden. The third phase we will determine the verification dose. And in phase 4, we're going to perform that verification dose. This process will establish the minimum routine dose required for the product. In phase 1 of the microbiological radiation validation, we'll need to determine accurate test methods for the device. These will include bacteriostasis, fungistasis, the bioburden test recovery efficiency, and, if needed, a Bioburden inhibitory substance screening. In phase 2, we'll perform the Bioburden method. We'll enumerate and characterize the Bioburden on the product. Here, the number and type of organisms present on the product are determined. This will be carried out on at least 3 batch sizes with 10 products each, using the test method from phase 1 product validations. In phase 3, we have a determination of the verification dose, using either the overall average Bioburden determined in phase 2 or the highest batch average Bioburden from the 3 batches tested. To determine the verification dose, we'll look up in the appropriate standard on the table associated with the method we're following. In phase 4, we're going to perform tests of sterility on the product. For method 1 approaches, we're testing 100 products. In the VD Max approaches, 10 products are being tested. That rounds out the phase validations. The next important aspect to remember here is the standards involved with these laboratory tests. The first one there is for Bioburden, the ISO 11737 Part 1. The next one is the sterility tests and the BF testing. This is in the ISO 11737 Part 2. Moving on, we have our sterilization dose and max dose and how these two are related. While the maximum dose is not a process validated under the microbiological radiation validation, it is an important consideration when selecting the approach. The minimum dose is what we're working with here today, and it's the microbiological substantiation of the sterile claim, while the maximum dose is based on product tolerance and required for performance specifications, i.e. the product functions as intended and is safe to use. Doing the max dose study early and before microbiological work begins is very beneficial. You can determine, one, that the radiation sterilization is the correct modality for the product, and secondly, it determines what the product's upper dose limit is, which can be critical in deciding on the optimum minimum dose, allowing a wide dose range as practical to give confidence for routine processing capabilities. Moving on, the radiation standards and the approach options. We have ISO 11137 Part 2. This is our sterilization dose, outlining methods for method 1, method 2, the VD Max 15, and the VD Max 25. The second standard that we're discussing is the ISO 13004. This expands on the VD Max approaches. It outlines 7 new approaches covering dose ranges from 17.5 up to 35 kilograms. Looking a little bit closer at the ISO 11137 Part 2 approaches. In this document, it has a long history as a standard in ISO, and it accompanies the other ISO documents, Part 1, Part 3, and Part 4, which cover all the other aspects of the radiation validation. In ISO 11137 Part 2, it describes the approaches available for selection of a sterilization dose and the technical steps required to determine that dose. The approach offers dose ranges from 11 up to 36.3 kilograms. There's also 3 approaches that cover a Bioburden range from 0.1 all the way up to 1 million CFUs. We'll be looking at method 1, VD Max 15, VD Max 25. It's also important to note that method 2 is another approach. It is used for dose setting using a fraction positive incremental dosing. However, it doesn't really follow the four main phases of the microbiological radiation validation as we outlined on the previous slides, and we will not be covering it in depth in this webinar. The ISO 11137-2 approaches. In this table, we can see the tables that are listed for determining your product verification dose. And in cases of method 1, our sal doses additionally are in those tables. It's also important to note that in method 1, there are two different tables, one to cover the low bioburden range below 1 CFU. So a little bit closer look at method 1 approach. Method 1 is a well-recognized approach where we're going to require 100 product samples for sterility testing, which can be a good option if your product batch sizes are large and the products are relatively inexpensive. The other thing to note about method 1, it has a large option for routine dose and bioburden on your product. The bioburden range, like we had talked about, can go anywhere if we're looking at table 5 from 1 to 1 million CFUs, and we'll use table 6 for less than 1 CFU to give us different dose ranges there. The other thing to note here is method 1 differs from the VDMAX approach in that there is no predetermined routine dose. Instead, you're determining the bioburden average value, and then you're going to look up on the tables that bioburden value determined in phase 3, the verification dose and the associated sterilization dose at your sal of 10 to the minus 6. So here in this table, we have a snippet from table 5 of ISO 11137-2. It gives the bioburden dose values for the method 1 approach. So what we'll do is we'll use the bioburden data we generated during the phase 3 of the radiation validation to not only determine the verification dose, which has under the column 10 to the minus 2, our verification dose, but we'll also determine the routine minimum dose, which will be under the column of 10 to the minus 6. You can see those two have been highlighted. And for example, if we look at an average bioburden of 1.5 on the table, we can see that the verification dose of 10 to the minus 2 will be 3.3 kilograms, and the routine minimum dose will be 14.8 kilograms. The next one to consider here is the VDMAX 25 approach. VDMAX 25 is commonly used and a go-to approach for validation for many different devices. It requires 10 samples for sterility testing during the initial dose establishment and for any ongoing maintenance, just like in method 1, i.e. periodic dose audits. This is a good option when you have small batch sizes or maybe the cost of the product is higher, since a lower number of samples will be needed for the sterility test compared to method 1. This is applicable to products having an average bioburden of less than or equal to 1,000 CFUs. From table 9, we can see that the bioburden can range anywhere from 0.1 up to 1,000 CFUs. Your final minimum sterilization dose will be 25 kilograms. And it is important to note that a SIP approach is allowed. However, you cannot use a SIP if your bioburden is less than 0.9 CFUs, but anything above 1 CFU, SIP approach can be used. Here on this table, we have a snippet from table 9 in the ISO standard. It gives you the bioburden ranges on the VDMAX, which go from 0.1 to 1,000 CFUs. From this table, we can use this to find the verification dose associated with our products. In those two designated red columns, we can see that you have low average bioburden and your SIP equal to 1 .0 VD max 25, you can read out what your KGY verification dose would be. In the green column, here you're above 1 CFU, and you can read the verification dose there. There is another column there, the SIP dose reduction factor, which, depending on where your bioburden lays, could be applicable. The next approach to consider is the VD max 15. This is often employed to validate products with low bioburden averages, and it shares many similarities with our VD max 25. It requires 10 samples for sterility testing during the dose establishment and ongoing maintenance. It's a good option for small batch sizes again, or high cost products, since there's a lower number of samples. Here our average bioburden needs to be less than 1.5 CFUs. So you can see on table 10, our bioburden range from 0.1 up to 1.5 CFUs. An important facet here is when we're determining our bioburden, we need to keep any multiplying factors during that testing as low as possible. This would be applicable when testing for aerobes, yeast, molds. A dual incubation approach can be used to give you an overall dilution factor of 1. That way you can calculate appropriately. This one will give you a routine minimum dose of 15 kilograms, and under this approach we must test the entire product, so a SIP of 1.0 is required. Here we have a snippet from table 10 of the ISO standard, which gives bioburden and dose values for the VD max 15. This is the entire table, and we can pull up our verification doses. There is no SIP reduction factor because a SIP is not allowed. So we just see that depending on our bioburden level, we can have our dose range for verification. Moving on in the ISO standards, we have ISO 13004 approaches. This document has a long history as an ISO standard, originally as a TS and then becoming an ISO document. And now we're moving on to 2023. This document extends the approaches available for VD Max. It has the same technical steps as a VD Max 25 or VD Max 15 approaches found in the ISO 11137-2. This standard offers sterilization dose ranges from 17.5 up to 35 kilograms, and there are 7 different tables associated with those dose verifications. Some additional information on the 13004. We had talked about the 7 different approaches, and here they are. So we can see the associated tables with each of those approaches within the ISO standard. And we'll use those tables to look up the bioburden, which will give us our product verification dose. The 13004 standard often offers our 7 different approaches based on bioburden ranges from 0.1 up to 440,000 CFUs. We can reference table 3 in the ISO standard, and we can map out exactly where those maximum ranges fall. For each of the VD Max approaches in the ISO standard, the SIP is allowed when your bioburden is greater than 1. However, you will not be allowed to use a SIP if your bioburden is less than 0.9 CFUs. The other thing that's important to note is that your bioburden average from each of the 3 batches must be less than or equal to the upper bioburden limits associated with that approach. So we can take a look here at a replication of table 3 from the ISO standard. This is the 7 different approaches for each selected sterilization dose. We can map out what the upper limit for the average bioburden is. So each of your 3 batches, let's say for 17.5, would have to be 9 CFUs or less. It's a very useful table for a quick reference to see what approach options are available based on your bioburden levels. That rounds out our overview there. We want to take a look at some approach selections and some working examples. Often we come into a radiation validation with an approach already decided for a product family. Some of the possible bases for this approach is maybe we've used this approach previously and we understand what's required to complete this microbiological validation. Or perhaps the approach is widely understood and talked about in the industry, kind of a tried and trusted method. For example, maybe a VD-Max 25. However, there are several factors that can influence or drive a requirement to change the approach selected. One of them being the effects on the product functionality, or maybe a discoloration is noted on the product materials above the planned minimum dose. Or possibly your processing range is too small or too tight. The range between your planned minimum dose and your product's maximum dose, and it would impact your routine processing capabilities. Another option is maybe the bioburden is greater than what's required for a selected approach. Or perhaps there's a need for a SIP where you hadn't tested SIP before. So let's take a look at some examples, and we're going to start here with example 1. Here, they had originally planned to use a VD-Max 25 approach on their product. During the Max study, the customer noted that the transparent portion of the product became discolorized with doses over 40 kilograms. This was not acceptable. They needed to see what approach could be used instead, using the dose options of less than 25 kilograms, as they determined that a dose range of 15 kilograms configuration was a bit too tight. They needed a routine processing range of 20 kilograms to be more achievable. They did determine that the product's bioburden was 19 CFUs. The approach options available with a dose of less than 20 kilograms, when we look at ISO 11137-2, the VD-Max 15, it requires bioburden levels up to 1.5 CFUs, so this was deemed not suitable. Method 1, we know, offers wide range of doses and bioburden levels up to 1 million CFUs. And when we looked up for a dose of 20 kilograms on the table, a bioburden of less than 46 CFUs was needed. With us having 19 CFUs, we determined that this would be suitable as it was below the required dose of less than 20 kilograms. For reference, off of table 5, here we looked up the 19 CFUs that we determined in our bioburden, and it gives us a routine minimum dose of 18.6 kilograms for our 10 to the minus 6 SAL. So our approach options with a dose of less than 20 kilograms when looking at the ISO 13004. The VD-Max 17.5 requires bioburden levels up to 9 CFUs. We couldn't use this method. However, the VD-Max 20 requires bioburden levels up to 45 CFUs. Seeing that ours was at 19 CFUs, we determined that this would be a suitable approach. In the table below, it shows the options that we have available to the customer based on bioburden levels of 19 CFUs. With a dose requirement of less than 20 kilograms. In this example, the customer chose to use the method 1 as based on the bioburden levels of 19 CFUs, gave them the widest possible routine processing window with 18.6 to 40 kilograms. It's important to note that the customer's decisions were also based on the following influences. 1, the product is produced in large batch sizes. The product is also inexpensive with a low manufacturing cost. Also, it was not a complex product from a testing standpoint of view. It had low handling requirements and low media volumes, therefore overall lower cost. Therefore, the requirement for 100 samples for sterility as part of their initial validation and any subsequent dose audits wasn't as significant in comparison to having a widest routine processing range as possible using the method 1. Our next worked example, number 2. This one, the customer originally planned to use a VDMAX 25 approach on their product. However, after testing, they've determined the overall average bioburden was 2,536 CFUs. As this was above the required bioburden levels for VDMAX 25, this approach was deemed not suitable, and a new approach was going to be required to determine the dose. The max dose for the product was determined to be 55 kilograys, and for this product's configuration, a processing range of 20 kilograys was acceptable. Therefore, a minimum target dose of less than or equal to 35 kilograys was required. The approach levels greater than 1,000 CFUs in the ISO 11137-2, method 1 offers a very wide range, as we know, up to 1 million CFUs. And if we look up our bioburden levels of greater than 2,536 CFUs, we can see that we get a dose of 26.4 kilograys. This was deemed a suitable approach as the method 1 fell into the bioburden range required. Looking at the ISO 11137 table 5 for method 1 approach, we can see that there is no bioburden value for 2536 CFUs listed here. So we need to use the next value above, which is 2600 CFUs. Using this as our routine minimum dose will be 26.4 kilograys to satisfy our 10 to the minus 6 SAL. In the additional approaches for 13004, we see up to 35 kilograys. We have 4 different approaches that we can use, covering a different wide range of bioburden levels. As we can see, up to 440,000 CFUs. However, when looking at this, we notice that the widest range would, at 27.5 kilograys, would be the optimal level to select, as this gave us an upper bioburden level of 5,000 CFUs, which would cover our 2536 CFUs needed. And in the table here, we can see that the customer had these various selections available to them. Ultimately, they chose to use the VDMAX 27.5, as this approach allows for a wide routine dose range of 27.5 up to 55 kilograys, and still requires just 10 sterility samples for the VDMAX approach. The customer's decision to use a VDMAX 27.5 was additionally influenced by the following. The product was produced in small to medium batch sizes. Additionally, the product was large and therefore complex from a testing standpoint of view. There was a high level of handling requirements, large media volumes leading to higher testing costs. Therefore, the requirement for 10 samples for sterility testing as part of their initial validation and any subsequent dose audits was a significant factor that, coupled with a wide routine dose range of 27.5, the best option for their VDMAX. Moving on to example 3. Here, the customer wanted to use a VDMAX 15 approach on their product, which would require a low routine dose needed for their product functionality. A max dose study required, and they determined that 35 kilograys was the max dose for this product. In this configuration, they determined that 15 kilograys was achievable, and therefore a minimum dose of less than 20 kilograms was required for this product. To use the VD Max 15 approach, it would require bioburden levels of less than 1.5 CFUs. When planning to use the VD Max approach, it's important that you must test the entire product, i.e. a SIP of 1.0. It's also important to keep your bioburden test multiplication factors as low as possible. This would include doing some sort of low dilutions on the product, sample filtration, the use of dual incubation on plates. Here we had determined that the overall bioburden average though came out to 3 CFUs during the study, and we determined that the VD Max 15 approach would not be suitable for this product. So the additional approach options available for 3 CFUs with a less than 20 kilograms, looking at the ISO 11137-2 method 1, again gives us a wide range of bioburden doses and doses. 3 CFUs gives a dose of 15.8 kilograms. This was determined to be a very suitable approach for the product. And if we look here, table 5 again, we have 3 CFUs and our 10 to the minus 6 SAL at 15.8 kilograms. The approach options for bioburden levels of greater than 3 with a dose of less than 20 kilograms in our ISO 13004. We have 2 approaches available. The VD Max 17.5 would require bioburden levels up to 9 CFUs, reference table 5, and then in VD Max 20, we could have bioburden levels up to 45 CFUs, reference table 6. Both of these would be suitable as they meet the criteria for bioburden and also the required upper dose of less than 20 kilograms. So in our table here, we can see the 3 approach options available to the customer. Ultimately, the customer chose to do the method 1 approach, as this gave the widest routine processing range as possible at 15.8 up to 35 kilograms. It's important to note that the customer's decision was additionally influenced by the fact that the product is produced regularly in large batch sizes. Also, the product is physically small. It's not complex from a testing standpoint of view. Low handling requirements, media volumes, overall low cost. Therefore, the requirement for the 100 sterility samples is not a significant factor, as opposed to getting the widest routine range that would impact their product functionality. Moving on to our last example here, example 4. The customer was unsure about what approach to use on their product. They did do a max dose study and found that the upper dose level of 55 kilograms for their product was achieved. Feasibility studies in the lab classified the product as a bit complex from a testing standpoint of view, and it was determined that the SIP of the product would be the best approach for testing purposes. For this product's configuration, a process range of 20 kilograms was acceptable. Therefore, a minimum dose of less than or equal to 35 kilograms was required. After doing bioburden recovery studies, 764 CFUs was the calculated value for the bioburden. In the ISO 11137-2, the VD Max 15 requires entire SIP to be used. So we found out right away this would not be a suitable approach. VD Max 25, the entire product item or SIP of 1 is used if the bioburden is less than 0.9. Our bioburden is not, so a SIP of 1.0 can be used if the bioburden is greater than 9 CFUs. So it was determined that the VD Max 25 could be a suitable approach, as the bioburden was greater than 9 CFUs, our SIP could be less than 1, and our overall bioburden didn't exceed the VD Max 25 requirements. Additional approach options, method 1 has a wide range again of options here. So the entire product must be used if the SIP is less than 9. However, a SIP of less than 1 can be used if the bioburden is greater than 9. It was deemed suitable since our product bioburden was greater than 9 and it's above the acceptable bioburden for use of a SIP. So method 1 is available. And for example here, we look at table 5 from method 1, and we'll have to round up again. Our CFUs of 764 is not listed on this table, so we go up to 800 CFUs. This gives us a routine minimum dose at 10 to the minus 6 SAL of 24.5 kilograms. Additional approach options in ISO 13004. All the VD Max 25 requires that the SIP of 1.0 is used if the bioburden is less than 0.9. However, a SIP of less than 1 can be used if the bioburden is greater than 9. These were deemed suitable as the product's bioburden is greater than 9 CFUs. So it's acceptable to use our SIP. When we look at the table here, we can see the different approaches available, and we've outlined their suitable options. Where the minimum dose of less than 35 kilograms is and a SIP of less than 1 is available. With the product's bioburden average of 764 CFUs, only approach options in the table from VD Max 25 and down were available for selection. In this example, though, the customer chose to use the VD Max 25 approach, as this allowed a wide routine processing range from 25 up to 55 kilograms, and with the requirement of 10 sterility samples. So some key takeaways from the approach selection. The max and min dose gives us our processing range. Consideration needs to be given to the product's acceptable max dose and the planned minimum dose to ensure that there's a sufficient dose range. This gives confidence in the routine processing capability for the product. Secondly, on your bioburden levels, they need to be considered when selecting approach. All the VD Max approaches have an upper bioburden limit, which will need to be met, or in the case of the method 1 bioburden levels, they'll be determined for the product's minimum dose. Product size and test requirements. Another important facet when selecting your approach, consideration may be needed to be given to products testing requirements, i.e. if a SIP is going to be required, the requirement to test the product using a SIP, not all approaches will allow or have restrictions on when a SIP can be used. The number of samples required for approach versus the testing costs. For complex products, for example, in method 1, you require 100 samples for test of sterility. A complex product may have a higher testing cost. Our last key takeaway here is our product availability. So test samples required for the approach selected may be a factor that also needs consideration. Your product quantities, batch sizes. In method 1, we're going to require 100 samples for sterility testing during validation and during dose audits. Production frequency is another facet there. And then the associated product costs if there's an expensive to manufacture product. Any or all of these can have an impact on decision on which approach fits you best. So it's important to select the correct tool for the job. ISO 11137 Part 2 and ISO 13004. Between them, provide multiple approach options to meet the needs of a wide range of product doses or bioburden requirements. Thank you. And for more information, please visit us at www.steris-ast.com.